Someone can start. Good morning to all. I just forgot today that is a second and third lecture that was shifted. So Prabhu sir has came and told me, and then I just recalled it. So I little bit late, 10 or 15 minutes from my end. Yeah. No, only one lecture. Yeah, yeah. Then after Prabhu sir is going to take, because I just forgot that is shifted. So in my hard copy, it was written by the third lecture. So I was waiting. I was taking tea, and on the way I come to know about it. So I just directly came. Yes. So we were talking regarding the colonial broth culture characteristic, right? So in that, we were till reached three important phenomena. Yeah, pellicle, sedimentations, and flocculent growth. Some basic parts we have just seen. For that, just I have taken one documentary that is related to. It is related to both culture characteristics, some on the plates. What are the colony characteristics will be there, and another it is related on a slant. Okay, on a slant, when you are growing certain things, what going to generally happens when we are going to have the slant will be prepared on a tube, it will be the surface will be somewhat concave in a nature. Okay, while preparing it, sometimes may happen that 2 to 3 things possibilities are there. That is not whole on the surface. If you are going to keep on the standing condition, slant is if you're looking to keep your media on the one condition. It's like a seems here the water bottle is there. Can you give that bottle to me? Yeah, attendance link I have pasted. Otherwise, I'm going to put again. It's like when I'm standing, this bottle is there. If this solidified media is there, what's going to happen? It's going to get solidified like this. But if I'm going to keep in the one condition, okay, near to the some racks are there, you can utilize certain things. If after a 20 minutes, 30 minutes, what's going to happen? It will be converted into a slant. What is the first requirement of preparing the slant on a tube? Such basic question is coming to in your mind. Okay, first is to increase the surface area. Here, the surface area has increased. Which surface area? Where you are going to work it out. Okay, the first important part is the surface area has increased. Second important part is going to happen that when you are going to streak, okay, it's become easier to streak on the plates. It is not a petri plates. You can open it and you are going to streak because in a tubes, when the search part is there, it's very much difficult to streaking. So if it is on an angle, at the certain level, you can utilize your wire loop for making a streaking over there. There you cannot do a spreading. Spreading is not possible. Okay, just if it is spreading is like that, you can put here just drop. It will be just spread. It is like you can put the drops like a water droplet is there. You can and you can just rotate. That can be possible, but there will be no proper development will be there. So two important features are there. We can have the surface area increased. Second one is what? We can easily do a streaking over there. Two important part for the slant. And generally, what is the purpose of this slant is there? That related to a storage. When you're looking to preserve certain kind of a microbes, it's really difficult to have the flask. 250 ml or the 1 liter flask you can store in a fridge, but it will take a larger space and area. If you can keep the plates, but plates also going to have the more areas to be get covered. So if it is the slant or the tube is there, very easy. Okay, you can make the replicas from the one itself. While the sticking, you can have the 5 tubes, 5 replicas you can store, and it can be stored at a different fridge, refrigerator. So it became easy. Sometimes happen that you have stored everything in one fridge. Power fluctuation has came, or something has happened. You are on a 1 week, like it is the holidays are going to come, right? You were not coming in institutions, and after 1 week when you are coming, the fridge is totally leaked because the ice and everything has melted. And when you open it, you will get surprised that everything is totally get degraded. Because afterward, what going to happen? Normal temperature may arise. Bacteria will start to grow. There may be a chances of getting contaminations by itself. Other microbes start to utilize their media. Sometimes it may remain, otherwise their viability may lost. Since the temperature fluctuations for the continuous, maybe their viability is lost. So we have to maintain the refrigerator temperature and everything. So such sort of things can be problematic. But the major important part is what? That is related to preservation. So on the slant also, the bacteria will going to show, and the plate, they are having some different behavior. In a culture, liquid broth is a different behavior, and in a tube on a slant, they are going to have some different behaviors. Okay, so behavioral part is just changing, so that we are going to just observe all the bacteria, all the colony, all the surfaces are having different. Yes, another important part over there, when you change the media, it is like a nutrient agar. You may use the McConkies, or you may use the blood agar or chocolate agars. With this also, their morphology may change. Okay, it is like the when you are taking the certain dishes, it is a sweet dish you are eating, your mood will be changed. Okay, you are taking the something different. Calories are there. If you are on a fast for 10 days, you are in hunger mode, then also behavior change. So always the media what you are feeding there, it's the most important essential part over here. So just one small documentary is there. I think you may have your mobile get on. Must? Yeah. Slant most important part is related to for the preservation. That's the main targeted part is there. Why we are going for the that is preservation. Okay, for the future study. So in a less space, if we are going to store the our isolate or the pathogens or any bacteria is there, it's become more convenient and easy. If the larger space is there, chances of getting contaminations will be more. As the space increasing, the chances that opening of the leakage of the plates can be possible. Sometimes may happen that you have kept the plates. Some other students are working on the same, you can say, refrigerator. By chance they have came, okay, they are doing their work, and they have started to just move here and there. While this may happen that you may have kept the inverted even a plates. It gets somewhat just little bit get exposed to the air. That's it. Other microbes are ready to cover that area. It's like currently the war-like structure is going in Afghanistan. Yeah, the Taliban and others have tried to acquire. Air is being exposed, all the plates will be get covered by the other organism. Right. UST militants were there. They have taken their parts. That is a well-known the phenomena is going on currently. Same thing is there in the real life, microbial life. Space is available. Other organism, it is opportunistic. It's not like that they want to grow, but they are looking to get that media. But they got exposed. The medium is got exposed. Air just circulated within inside. The problem is struck. Yes.
If so we keep in refrigerator, let's like come down into a normal temperature of so if it is not disturbed, then the other microbe won't get like contaminated on that. But if it is tightly and it's on requirement, if we are close to leak out.
Yes, if you are keeping the plates, you can keep the same plates in the any cupboard also. But what's going to happen? They are going to start to utilize that media. We have to stop their metabolic activity. The reason is what? If they are active, they are going to grow and grow and go and start to utilize. The day will come. After that, the media is totally exhausted. Then after the starvation will start, and the microbes are going to retain their growth, and they're going to die. There may be revival formations is there. If you transfer them to the next media, they can. There is some limitations for all the microbes. Yeah, another important part. Do you know about the apoptosis, programmed cell death? Have you heard about it? Huh? No, never. Okay. Have you heard about the cell cycle? Very good. Broadly, we can classify interphase, metaphase, M phase, and interphase. Right? There we are starting with the G0, S1, G1, like that. Certain phases are there. Okay, then the resting phase and cells is going in the metaphase. Mitosis is going on, two cells are going to get converted, daughter cells. It is the continuous process. During this time, the cell knows, shall I go for interphase or a rest in a life? So they are going in the program cell death. In between also, they may reach into G0 to G1, resting phase will come, they are halted. Shall I go for the S phase or not? They may abort that S phase, program cell death is there. That means they are going to kill themselves. Programming is there. When it is not, it is like an uncontrolled growth. Cancerous part is going to happen. So all the eukaryotic cells measure are having that characteristic part over there. But bacteria only learn to grow. They never learn to get died. Just to grow. If when they are going to get die, there is a no nutrient availability. They like to survive. Okay, in which format? They may form their spore. All the cells cannot produce the spores. They are keeping they will limit their metabolic activity and they are going to control a level that they are going to get survive. But after one hungerness, okay, nutrients depletions, water scarcity, two major important factors to survive in a life, is going to get exhausted, bacteria is going to get dead. So you can preserve, same thing, but preservations is they should be get survive, metabolically remain active, and when you are doing the subculture, they start to grow. Okay, it is like the giving the punishment in a prisoner. It's not going to kill. Okay, some food is been given at least in a day. At least basic requirement is given to the prisoners also. Okay, it is like if you are going to feed something, they are get processed. Okay, after process, each having their excretory system also activated. So sometime in a prisons, you are knowing that it has been attached with the toilet. Okay, it's a basic need. It's a part of a human rights. One, second is related to their basic physiology. Otherwise, he is going to die or she is going to die. So we have to manage. They should realize that they are get punished. So we are keeping that metabolically them active. Okay, we are not going to give any preservation to our microbes, but we are looking to utilize in the future. So they should remain in active form. We are not going to give the full hungerness to our prisoners. We are going to feed them. So microbes should remain metabolically active and both. So we are looking to give the slant. Less space is available. Okay, if the prisoner is given the larger room, there is a chance they can roam, they can find the place. The smallest area is given, okay, 2/6 maximum or 3/6. You can understand the square foot. Your seti palang is there. Maximum 3/6 palang is there, or 2.5 and the multiplied by 6. This space is just given, so you can just locate, relocate. You can sit down, stand. You can sleep. Space is prepared in a way that related to the slant. Okay, so that's the base part. So just open up this having the this audio is having some let me share along that with sound. Small documentary. Okay, it's about a maximum of 4 to 5 minutes only, but you will like it.
Basic microbiology, a laboratory manual, colony morphology, or description or characteristics. Purpose: to separate bacteria into taxa, to identify bacteria. Diagnosis: cultural characteristics are
Why we
determined
required?
using nutrient agar. They are viewed by
Characteristics to be get
lenses
understood.
or
That
best
he is
by
going to
stereo
say first.
microscope due to 3D image.
To separate the
Mobile
bacteria,
phone cameras
cultures are determined
provide
using
good
nutrient
images to
agar,
judge
view it,
morphology. Types of
for the
medium.
document
4 types
purpose.
of nutrient
Major
medium
things are
to
we are
determine
looking to
characteristic. Nutrient agar
understand
slant,
their
nutrient
characteristics.
agar plate, nutrient
Other
broth,
various places.
and nutrient gelatin.
Agar slant,
Reported
agar plate, broth,
characteristics: form or
nutrient
shape,
gelatin currently
abundance,
that is
consistency,
a
optical
substance
density,
available
and
for
odor.
the solidifying
Sniffing
agents from
may
the
cause disease. 1.
plant source.
Nutrient agar slant.
Sorry, animal
A.
source. Nothing else.
Forms of a single line streak on nutrient agar slant.
આને slant કહેવાય.
Or thread-like. Ecannulate with
આ દેખાય છે
spines.
આડું તૈયાર
Beaded.
કર્યું હોય.
Effuse.
ટ્યુબને તમે
Arborescent.
આડી કરી.
Tree-like.
તમને
And
બનાવતા
rhizoid.
શીખવાડીશું slant.
Filamentous.
સ્લોપ આપણે
B.
કહેતા
Abundance
ગુજરાતી.
of growth
સ્લોપ
of a
બનાવવાની
single
વાત
line
છે.
streak on nutrient agar slant.
So it's just
No
showing the
growth.
some characters.
Slight.
Moderate, slight,
and
no
large.
growth. There is a
C.
large growth will be
Consistency
there.
of growth of a single line streak
Growth
on
consistency.
nutrient agar slant.
Dry, butterfly,
Buttery,
mucoid.
and
Just
mucoid.
I'm going to explain. Just
D.
see
Pigmentation.
that documentary
Some
part
bacteria are
is very
chromogenic color
in a
medium.
speed and hurry.
Green,
Within
red,
2 to 3 minutes,
or
you're going to
yellow.
deliver a lot.
D. Optical characteristics
Sometimes
of the growth
pigmentation.
of a single line streak
Okay,
on nutrient
that is
agar
optical
slant. Opaque,
characteristic.
with no light
Slant is
transmission.
before to you.
Translucent, with
You may
partial
be
light
able to
transmission. Or
See that the light
transparent
can be
with full
passed
light
somewhere it is
transmission.
not.
Colony
So it's related to
characteristics on
opacity
plate.
even.
Form:
Now the plates,
circular, irregular,
whether it is
filamentous,
circular,
and
the
rhizoid.
colony will be appeared in different
Elevation:
formats.
raised,
Rhizoid,
convex,
filamentous, irregular,
flat,
circular.
ambulate,
Elevations,
and
when your
crateriform.
plate is there, you are going to
Margin:
view from the
entire, undulate, or wavy
angles
indentation.
from the
Filamentous,
maybe
serrate,
180 degrees.
or
Raised,
tooth-like,
convex,
and lobate
plate,
with marked
embonod,
indentation.
catrophoids,
Size:
margins,
pinpointed, small,
surface is hilly,
moderate, or
having the
large.
tree-like, rhizoid-like structure.
Pigmentation:
It is like a playing
red,
with the microbes.
green,
Okay.
yellow,
Or
or white-gray.
size, sometimes moderate, pigmentation. These are the some 4 to
Nutrient
5
broth:
basic
appearance of
parameters
growth,
are there.
uniform,
On a
flocculent,
growth, nutrient growth,
Delicate.
just already we have seen.
and
Uniform
sediment.
flocculating
Prepare broth cultures of
pellicles.
Pseudomonas aeruginosa, Bacillus cereus,
When can be possible? After a
Staphylococcus
like
aureus,
76 hour,
Escherichia coli,
26 hour,
Mycobacterium
can be
stigmatis.
observed.
Provide each group with 1 nutrient broth, 1 plate, and 1 slant for each organism. Inoculate broths,
So he's
plates,
showing
and
about the
slants
different
as
organisms.
described in lab
Pseudomonas,
2.
what will be their
Briefly,
characteristic? For
inoculate
others, what will
slants
be
with
their
a sterile
characteristic?
needle in a single line streak of each of the cultures provided, starting at the butt and drawing the needle up to the center of the slanted agar surface. Streak plates with a sterile loop to produce isolated colonies. Inoculate broths using a sterile loop for each organism. Shake the loop a few times to dislodge the inoculum. Incubate all cultures at 37 degrees centigrade for 24 to 48 hours. Use this form to report growth on slant. Refer to previously shown figures for comparison.
So he's
Use
like
this form
what could
to
be
report
there?
growth
It is
on
like in
plates.
journal.
Refer to
When you are
previously shown
going to
figures
do
for
certain
comparison.
parts, such tables
Use
is
this
required to
form to
prepare.
report growth
Okay.
in broths.
Basic
Refer to
characteristics.
previously shown figures for comparison.
So, not now, later. જ્યારે આવશે ત્યારે હું કહીશ તમને. That time we are going to grow. So reference part is gone. So it is related to what? A slant to be get prepared. So in the next experimentations, we are going to have the one plate and another maybe a slant. Okay. Keep ready. I'm not sure. We are just going to otherwise like on a Wednesday or a Thursday. For a one day we may have the change. Maybe I may take your lab tomorrow. Okay. On a Thursday it will be a chemistry. Some work is going on. We are going to finalize. So just be ready for the tomorrow. So we may have some directly practical sessions for the chemistry. So what will be our targeted part? I think already you have done the calculations. Groups will be prepared, right? So we are going to have the solid media that is on a plate. Plates were already ready. You have prepared the tubes even, right? That part was ready with you people. So what you are going to do directly? You are going to prepare directly our mediums. Okay. So in that, two possibilities are there. you may pour from the beginning in a tube itself, because you are learning first time or doing first time, like you have prepared the nutrient broths. Otherwise, what's going to happen? That as you are taking out the broths out of the autoclave, you have to start to pour very faster. You are just arranging and doing it, start to get solidified within the flask. So it will be better for us and you that you may transfer about 5 ml, 20 ml, what will be the instructions will be given to you according the tube size we will finalize. Okay, that amount should be transferred in a tube itself. Okay, and that tube you are going to put in the autoclave. So when you are taking out from the autoclave, you just need to manage in an angle itself. Okay, so it's became more convenient and easy. And yeah, plate we have to autoclave. Pouring will be done in the plates. There you cannot. If you are going to put the everything in a plate, in autoclave it will start to boil. It will be come out. So it's always not feasible. So over there we cannot do such things.
Basic micro.
Okay, so this one is a slant. Some basic characteristics are being shown. There is a fill form, ignolate, beaded, effuse, herbosant, rhizoid. So what are each? It is nothing but like some different characteristics is being observed, like rhizoid-like structure is there. Sometimes the irregular different patterns are being shown. So it is the some characteristics of the certain bacteria. E. coli, Pseudomonas, Salmonella, some different organisms going to show their different characteristics. Why it is required to understand that when you have preservation is done, by visualizing on a second or third day, you can at least confirm that my bacteria is remain in pure form. You may store it, and after 1 month or 1 year, you are going to take it out the slant for doing the experimentations. And when you start to do experiment, you may say that the results are not coming as it was desired, because on the day you have transferred, it is already contaminated. So by just observing, you can say that whether it is in quality, maintained or not for the preservation purpose. So this characteristic is required. This filiform, continuous thread-like growth with a smooth edge. Whether it's relative, continuous thread-like growth with irregular edges. Here it you can say very smooth. Wherever you have made the streaking, it has not diffused or moved. It is just one line. Another is like a irregular. It is like some people when going on riding the bike, some are straight just on the one lane, another जी रहा हूँ मेरी अपनी जिंदगी पहला मौज में रखते हुए. So it is like something different. Okay, then the beaded non-confluent semi-confluent colonies means what? They may sometimes you may find the some species, some bacteria will not grow. I don't want to grow. Maybe it's happened, so it get diffused. So it is like riding the bike. જીને લગાહો પાછું એક વચ્ચે કઈ કેબિન આવી ગઈ કે ભાઈ સબ આવી ગયું, થોડોક નાસ્તો વાસ્તો થઈ જાય પછી હલાવશું. So journey is recorded. So you have taken the break. It's like that. So you have taken the pause, bacteria growing. Okay. So it is that. Diffuse. Yeah. Thin spreading of the growth. Very thin. So it's a different, some characters are there, how the bacteria will start to grow. Absence, tree-like growth. And another is like a rhizoid. Spreading. Okay, spreading like a rhizoid. Sometimes you may have seen such growth on your bread or some butters. If the stale food is there, on that you may have, it is related to fungi especially, not for the bacteria. But you may see in the some fungi are growing. Okay, same it is like that. Rhizoid-like colonical portions will be observed. So just it will be some characteristics, maybe it's like this. You are already knows that this bacteria I'm working. So it's become easier for you to understand their physiology. You can store it or you can preserve it for a longer time. If such growth is not there, then you cannot store because it's already contaminated. This is some basic characteristics. So why we are going for the slant preparation? Most important related to preservation. Another is to increase the surface area. Okay, and the sticking is more convenient. Okay, then the some are the slant are being prepared for the I will discuss about during the preservation. Okay, some slants are not being prepared in a slant. They are just prepared in this format. Surface is flat. Okay, so it is the tube agar. It's a plate agar. It's a tube agar. We are not going to call as a slant. Okay, tube agar is there. What we are going to do when we want to transport, I have shown about it, right? When we are going to use the needle, okay, our nichrome wire will be needle. What we are going to do? Just doing and piercing in the middle. Okay, so this piercing is done on the tube agar. It will be in that condition. So few microbes just inserted, and they medium will be having the less metabolically active and the less nutrient. So microbe will start to grow very restrictedly, and during that time, you are going to transport from one place to another. That's the major our agenda. For preservation, so many techniques are there that I will discuss in the separate topic. Okay, so till this concept is clear related to the slant. Slant સમજાય છે? Why we are preparing the slant first? Okay, what is the importance of the slant? Okay, getting? Anybody having any questions, any doubts, or not? I hope it's clear. Fine. Next point is related to colony. So basic 7 to 8. All the literatures are listed. Some are going to list up to 5. Some may list up to the 8. Some literature are showing up to 7. So I taken from the various places. The thing is what? Some maybe get connected with one to another. Okay, it is, you may find that it is the same, but some people are describing same thing in different words also. Okay, so let's see one by one. Colony, say, it is like a circular, irregular, punctiform, that is a tiny, filamentous, rhizoid, etc. So here we can see very easily. Some are the very circular. It is like a when you are having your break. Okay, right now or in a schooling time, when you are in a garden, 3 to 4 students are get together. Some are 8. Someone is coming again. Please give us some little space. થોડીક જગ્યા આપને એટલે જગ્યા આપે ન આપે. બરાબર છે, પાછળ બેસી જાય. So they will remain in a group. So it's became what? Irregular. They want to be remain as circular. Sometimes space is not there, they become spindle. Round banai nakho. Okay. If the get together is there, sometimes in a picnic time also, some family members are all to going in a place, right? So they want to enjoy by themselves. So there may be 1 or 2 group may happen. Elders are sitting, ideally. Okay. Children are playing here and there. Okay, it's like a flocculus, formation of flocculus. All the ladies will be start to move in that direction. It's like a dinner party at night. Okay, so they all are having their own space to get work. So it is like same. Some microbes will follow rhizoid filamentous. It's for them it is a party. Okay, so all the groups are going to have their own way to grow. Okay, but their characteristics will be remain fixed generally. So shape will be there, circular, irregular, filamentous, rhizoid, spindle, and many. These are the some limited name is given. Okay, sometimes they are following here and there itself. Okay, they are the given, there's a limited shapes are name is given. Sometimes you may observe something different also, but you can at least classify that is irregular, regular, irregular, round. Some basic shapes are there, and these are general characteristics of the microbes. If you are going to see the E. coli or the B. mega, generally their growth will be in round. Okay, they are growing in a from the middle part. The cell is there on a plate. They are going to grow in a round itself. Then the elevations from the side. From the side, you are going to observe. Okay, one is always flat. Some are raised, convex, fulminated, and the umbonate. Okay, it is like a some, you can say like a pyramid, or you can say the mountains are start to grow. Some are highly convex. smooth round will be there. Some are having the highly elevated pulvinage. Okay, so these are the some basic characters. And what going to happen? Sometimes they may rise in a way that over that area easily you can see. And not fungus, it is a bacteria only. Okay, related to the bacteria we are just talking. Okay, all are. So raised, just raised, it is not in round shape. So such characteristics are possible. Yes, you have to remember this name, at least few. Okay, so what are the characteristics these are there? Same question can be asked. 5 or 6 mark, 8 mark, colony characteristics. It is very easy, but you have to remember certain points. You need to draw, yeah. You need to draw it. Whenever I already told, my paper will be there. Okay, 50% is given attention always to the figures. Okay, that is the for most important part and should be labeled. Okay, out of 50, 50% is for the labeling, 50% for the drawing. How good you are or articulated, that is the very well worth. Sometimes some people has drawn. We have to write that it is a camel or a dog. Otherwise, you need to predict that it is something. So it should be appear with certain points. Then the margin. That entire margin from where? From the outer part moiety. It is limited to size shape also. Then the lobate, eros, filamentous occur. So many ways is this possible. That a margin outer part. Okay, it was raised. Now the round is there or not, fully circular or not. So there may be some are entire, some are the undulate, lobate, eros, filamentous, curl. You will observe it is like a preparing a rangoli. Articulations will be there. Okay, it's using the some flower or so many things you can possible. So it is generally considered in irregular shape, but you can have the definite shape named by these are the some of the groups I have studied and they have given the certain names to them. Then the size. Generally 4 to 5 mm is the normal. Okay, but the group of microbes are get together, then the size may change. It is required to a single colony forming unit should be there. If it is a lone growth means what? All the whole plate is fully covered. Okay, so here you can observe certain few bacteria. Okay, some are the colored even. So here one of the example is given the 4 nanometer or the 4 mm. You can have same plates. You can keep your here. You can keep your scale to measure their diameter even. Otherwise, what you can do? Same plates can be kept into a colony counter meter. Is there? Just already I have shown to you. So it's going to take the one pictures. By taking the pictures, it is going to say that how much amount size will be there using some software. It's nowadays very easy. Otherwise, we have to use some measurement like a scale. Or else another way. Maybe you have used in a physics. I'm not so sure much about vernacular or the some meters were there for measuring the size of some bead or something is given to you, or the screw. What will be their diameters? So such part may have used in that way. So such also been utilized. So you are going to slowly open that part. Here it is the below to it. It is not going to fit within inside. So some are the measurements the people were using earlier, but nowadays software-based analysis is more easy and convenient. So just I'm ending here. Some other parameters are also there. Here some example is given related to E. coli, some other pseudomonas or the B mega. So you can see that some organisms are smaller, some are the very much larger. When will be the growth will be given, they will spread. It is like you are sitting or you are just maybe it is in a like you are going in a train. Okay, you can sit itself so other people can accommodate. Some people are going to sit in a way that putting their legs on the seat, they will cover the whole seat. Okay, it is up to you what you like. So some bacteria will develop their colony in that way. Okay, they remain restricted for all time. But if you are keeping the incubation period, then the growths will be higher and higher. They are going to cover. It is not restricted. Okay, because they are here. Media is available, so they will grow. But it is a limited. After their limitations, they will not start to grow directly because their movement is not that faster. So such part we are just not looking to do understand. First, what we are looking to understand here, it is the general incubation period, 24 hours or the 48 hours or 72 hours, maybe a 2 to 3 days. Within that day time period, what's going to happen with my plants? Clear up to this? Some other characteristics are there that I will take in next lecture. I'm going to put the attendance link again. If it is some people has not put, you can do. So put the attendance within time period. Okay, it is not like the you may come to not 11 o'clock night and you are putting the attendance. If it is there, I'm not going to take it for the attendance. So within time period, in the lecture time, it is better. Maybe it is 5 or 10 minutes I may allow. Okay, it is the 8:30 to 9:30 or 9:30 to maybe or 10:30, whatever the lecture time is there, up to maximum 10 minutes. Okay.